right, so my name is Danielle, this is Brody, we have Camila and Kevin, Abby, Sydney, and Sarah also contributed to the presentation. So our team is focused on microplastics. So here we have an outline of the presentation that we'll be sharing with you today. So as our population increases, so does the amount of trash that we produce, specifically plastic. And that plastic ends up in our water systems like lakes and rivers, and it eventually has detrimental effects on our ecosystems. So not only is this a growing and global issue, we can also see the effects locally. In a study by Erickson and colleagues in 2013, they found that 43,000 pieces of microplastics per kilometer squared were found in the Great Lakes. So overall, we're hoping to investigate these impacts of the microplastics and figure out preventative solutions to mitigate the effects. So recalling back to our proposal presentation, we discussed our 3 research questions, and today we'll be going over the first 2 briefly. And to help us in answering these questions, we spoke to some experts. So let's take a look at what they had to say.
As everybody talking about marine microplastics, why is nobody looking at microplastics in the freshwater systems?
How are microplastics or plastics getting into Hamilton Harbor?
What all of the direct and indirect consequences are going to be.
A lot of the changes may not be reversible, but we can mitigate and counteract and make positive change, yes.
I think there's kind of 3 main issues us as consumers. It's the systems and infrastructure we have for our recycling, and then it's the manufacturers and producers.
When you think about all those rivers that are covered in plastics, I mean, it's very obvious that this will have negative impact. But then I'm living in Norway, and Norway is very clean cut, and then the effects might be much more subtle and not that visible.
We find a lot of microplastics and plastics along our shorelines. Plastic can enter our waterways through runoff. Plastics do wreak havoc on our native species. Almost 80% of the plastics in the Great Lakes can be found in the digestive tracts of fish, mainly in the form of microfibers.
So there are species that are easily affected by changes in water chemistry. It can definitely lead to feeding behavior changes. So I think the alteration in feeding behaviors would then change food web structures based on the behavior side of interactions.
Impacting our ecosystems, being consumed by different aquatic life, which was accumulating across the food chains. It's a lot harder to clean up microplastics as opposed to plastic bottles in our lakes or rivers, for example. So I think we're going to need a lot of innovation.
Alright, so the focus of our first research question was on the effect of microplastics on freshwater aquatic ecosystems. And this is where we were able to look at a bunch of case studies that explored this topic to show us how serious and significant this issue really is. So we grouped the first set of effects as molecular and cellular effects, in which we included gene expression, cell signaling, oxidative stress response, and antioxidant mechanisms. And here is just one of the many case studies that we looked into by Wright and colleagues, where they looked at microplastic consumption by lugworms, which increased their gut residence time, increased phagocytosis, decreased lipid reserve, and decreased overall total energy reserves. Now, in the graph beside it, this also supports the idea that the percentage of phagocytotic cells were increased, which has been shown to increase stress response in these fish species. So the next effects that we looked at were systemic effects, under which we observed digestive, immune, and neuromodulator functions. And an example of a case study we looked at showed by Franzaletti and colleagues in 2019, it showed different content of microplastic in different tissues in this fish species, including in gills, gut, and liver, showing that it spreads all over their bodies. The initial effects that I talked about eventually gradually build up to more serious effects that show deficits in locomotion, growth, reproduction, and feeding. And we grouped this as apical endpoint effects. And in a study done by Che and colleagues in 2018, they looked at exposure of microplastics to these fish, and they looked at their locomotion. So in these two different fish species, the total distances traveled in 30 seconds varied. For this one, it was significantly shorter. Meanwhile, for the other, it was slightly longer, showing that microplastics can have different effects on their normal locomotion.
For our second research question, we wanted to look at how the implementation of microplastics in freshwater ecosystems can influence the dynamics of the food chain and the overall ecosystem stability. A case study by Farrell and Nelson wanted to look at how microplastics are transferred from one trophic level to the next. To do so, they fed microplastics to mussels, and then they fed those mussels to crabs. After 24 hours, 15,033 particles of microplastics per milliliter were found in the crab's hemolymph, and after 21 days, 267 particles per milliliter. Researchers also found microplastics in the stomach, hepatopancreas, gills, and ovaries. This study was meant to highlight the natural transfer of microplastics from one trophic level to the next. Microplastics also affect the abiotic factors of ecosystems, such as water and sediment characteristics. We found in our research that microplastics affect water translucency, the sedimentation rate in the water body, and the thermal conductivity of the sediment. Now, these influences will be discussed further in our report, but these changes can affect the ecosystem dynamics between organisms. Besides the direct effects that plastics have, they also have an indirect effect on ecosystems. Through different disposal rates, microplastics can come in contact with environmental contaminants present in, for example, wastewater streams and landfill leachate. As a consequence, these particles and compounds can mix before, during, and after their intended use. By this process, a seemingly inert and non-toxic particle can become a carrier of toxic compounds. Beselling and colleagues suggested that microplastics influence the inter- and intra-species signaling as an integral component of aquatic biocenosis, regulating predator-prey interactions, as well as population and community structures. Now we're going to look at a video showing potential solutions to these problems.
In societally speaking, change happens at the behavior of the individual. Policy change too, but I think policy comes from the changes of individuals that want to see that change.
Humans in general have become a lot more of a disposable society.
Right now, McMaster's current waste management system, I would say, is very strong. 2 years ago, they implemented composting on campus.
Doesn't matter if you have a million compost bins, if it's filled with plastic and garbage.
McMaster as an institution doing great. McMaster's community, not so much. There's still a big knowledge gap there.
Reduce, reuse, recycle is in order. You should be reducing the amount of plastic you're purchasing, reusing what you can, then you should be recycling, but recycling properly.
Refusing. They offer you a straw, they offer you a container, say no. I have my own.
The world, Gaia, Mother Nature, however you want to describe, is more plastic and resilient than we give it credit for.
Going zero waste, definitely attainable.
We need a culture shift on campus. We need to be telling students that this is important.
There's still a long way to go.
Our team biodiverCITY went on a tour of the Hamilton recycling facility, where Mr. Dong showed us firsthand how the process of recycling happens, as well as many other resources and videos that were useful for our project research. Matters into our own hands and organized a shoreline cleanup of Princess Point in Hamilton, where we collected about 25 kilograms of waste. We then decided to do an audit at McMaster, where we analyzed the contents of recycling bins to see if people were recycling properly.
These are compostable.
And then we do have cars. So really, from 2 garbage bags, only 2 items were actually recyclable.
We need more compost
Yeah, we need them where people can actually see them.
This is fun and should not be thrown out.
So to follow up on this, we asked McMaster students how they would rate McMaster's current waste management and sustainability practices. On average, they gave McMaster a 3.1 out of 5 stars, which is a very average rating. And then we asked them what practices they'd like McMaster to implement to help improve this score. Majority of people said that they'd like to see improved recycling signage, additional compost bin, and more educational recycling campaigns on campus.
For our integration, we decided that we should focus mainly on the biotechies and Darwin's angels to integrate with, as we all are sharing an aquatic ecosystem. As well, we have the other three in our handout, if you want to have a look at those. For the Team Biotechies, we wanted to incorporate the ideas of nanotechnology filtration into our own current systems that are available on the slide here. The sediment microplastic isolation unit is already 96% effective at extracting microplastics, but it could be even better with nanofiltration, and this nanofiltration also could be taking out the estrogen if we work together. Some other technologies that were also mentioned, bioremediation using microorganisms. So in image A above here, we have PET, which is polyethylene terephthalate, which is plastic, being eaten by a microorganism, Ideonellus sachinensis. Now, this microorganism has, the sole carbon source of it is this PET, as seen in A. In B, this is a microscopic image that shows the PET film being degraded by this organism. And C, this is a time-lapse sort of deal of the weight of the 60 milligrams of PET being degraded over 70 days. That's all it takes, 70 days for these bacteria. Now, they could also implement biodegradable plastics into our solution, such as PHA, which is degradable in aquatic environments.
Now for integrations with Darwin's Angels, their group is focusing on consumerism of resources and how that affects feeding, migratory, and reproductive behaviors in aquatic species. So our group was able to form linkages with them, as we found that microplastics actually also have effects on these specific behaviors as well. So in a study by Cole and colleagues in 2013, they actually found that zooplankton that ingested microplastics had an overall reduced dietary uptake of any sort of food. And as we see, the microplastic is highlighted in fluorescence there. And also, there's evidence of decreased reproductive output, for example, in a study where they assessed Daphnia magna. And then there's also migratory effects. As Camila mentioned in the study previously, locomotion can be impacted, and that can also have effects on the migration of fish. So our group also is taking a look at a few case studies. So Darwin's Angels is focused on heavy metal toxicity leaching from agricultural or industrial practices. So our group hopes to take a look at the case study of the Guyu e-waste dump in China, where we'll be focusing on the improper management of electronic waste and how that leaches into, how the metal toxicity leaches into aquatic systems that way. And then we're also taking a look at the Great Pacific Garbage Patch. So although this is a marine system, it'll still give us a lot of insight, where basically it's a great representation of the overconsumption of resources as well as the mismanagement of the disposal of these resources. And there's also evidence of microplastics within the patch as well as heavy metal toxicity, hundreds and thousands of tons of waste from fishing units as well as offshore oil rigs connecting with Darwin's Angels as they're taking a look at overfishing and fossil fuel consumption as well.
Finally, we'd like to take up the activity. So, who had the plastic bag? That was this group. How long do you think it takes to degrade?
500
200.
500, 200?
Yeah.
Those are big numbers. It's actually roughly 20 years.
If you take off the last zero.
Yeah, exactly. Only by one factor of 10. Now, styrofoam. Who had the styrofoam plate? What do you guys think? How long?
10 years?
10 years, okay. It's not quite 10 years, it's actually 50. It takes a really long time. Now, for microfibers,
I'm gonna
t-shirts,
say 100.
100. That's actually a really good guess because it takes 200.
Wow.
Yeah, congratulations. Who had the plastic straws? TAs and Genrec. What do you guys think?
Say like 500.
500 and?
100.
Okay, you could have copied their answer because it's right. Very good job. And now who had the plastic bottle? You guys did, right?
Yeah. 1,000.
1,000.
Big money.
You looked that up, didn't you?
No, I didn't.
1,000 years.
Wow.